Our first speaker is Annemarie Wingard. She's a professor for molecular toxicology at the National Food Institute at Denmark's Technical University. She has published more than 110 scientific papers and is coordinating a number of European projects. The biggest one, Panoramics, just ongoing, project where BFR is proud to be part of as well. Annemarie will be talking on mixture risk assessment in the past and in the future. Thank you so much for joining. The floor is yours.
Thank you, Philippe. I would like to congratulate BFR with the anniversary, and I would like to thank for the invitation to come here and talk about one of my favorite topics, which is mixture toxicology. I'll try to give you a brief overview of the state of the art of the research field of mixture toxicology and also try to look a little into the future. So the vast majority of chemicals, they occur as chemical mixtures. So it's the norm rather than the exception. Then humans and the environment are exposed to chemical mixtures. And a growing body of scientific evidence tells us that chemicals, they act together to create combination effects, also at realistic exposure scenarios. And we know that some of these co-exposures, they can affect human health or the environment negatively. So in the current risk assessment system, it's usually the norm that one chemical is risk assessed at a time. It's done slice by slice, and often mixture effects are not taken into account in the risk assessment. But we really need to do this in the future. So one of the challenges for implementation of mixture risk assessment is to find solutions how to address aggregate exposure to the same chemical when it's regulated under different pieces of legislation. So in the current EU legislation, it does not provide for a comprehensive and integrated mixture risk assessment across legislations. And this is something which is needed. It's needed to find solutions for this in the future. Yes. However, there are exceptions where the EU legislation takes mixture effects into account, and some examples are shown here. For instance, for dioxins and related PCBs in food and feed, the mixture risks are taken into account. For certain phthalates in articles, there's also regulation in place. For pesticides in food as well, and recently for PFAS or fluorinated chemicals were assessed because of on the basis of their mixture risk. Then, as it's not possible to test every imaginable mixture, it's important to ask, is it possible to predict the effects of chemicals? And to quote Barack Obama, yes, we can. If we have knowledge on single compounds, on hazard and exposure of single compounds, we can, in most cases, predict the mixture effect. And that's really good news for the risk assessor, because it will enable, that we can actually risk assess chemical mixtures in the future. And I'll give the background for this, but first I'll just mention these two very fundamental different approaches when you are designing mixture studies, depending on the purpose of your mixture study. If the purpose is to test if or how the chemicals are interacting in the mixture, then you can perform this top-down approach where you select chemicals and doses based on their effect or mode of action, can be model compounds, and you perform those response curves of the single compounds and also of the mixture, and you can compare the observed mixture response with that predicted by mathematical predictions. And this is what I call the top-down approach. Then you can use the bottom-up approach, where the starting point is, it can be realistic exposure scenarios where you use human biomonitoring data, for instance, to design a mixture that you prepare and test in your experimental system. And a sub-variation of this type of mixture study is to test complex real-life mixtures, for instance, extracts from human blood in your system of interest. And I'll give you some examples of both of these types of mixture studies. First, to the top-down approach. Here shown some examples where studies have been undertaken with the purpose of showing how or if the chemicals are interacting in the mixture. And the data from the results from these studies show that the dose addition principle provides a really good approximation of predicting the observed effects. It has been shown, for example, for nibble retention in male rat pups that have been exposed during gestation, or it has been shown for micronuclei formation in cells, for vitellogenin induction in fish, or for androgen receptor antagonism in cells, and in many other experimental systems. So it's the norm rather than the exception that chemicals are acting additively when they are mixed. And synergism and antagonism are only seen in rare cases. Even if chemicals are only acting additively, they can still have a marked mixture effect. And this is what has popularly been called something from nothing, which has been observed in various test systems. So the single compounds do not provide any effect, but when they are mixed, they cause a marked effect. And this was first shown for estrogen receptor activation in vitro many years ago by Kortenkamp's group. Later, it was shown in our lab by in vivo when we measured shortening of anogenital distance in newborn male rat pups. And very recently, this was shown in zebrafish, where 8 compounds were tested for malformations in the zebrafish. So even if chemicals are only acting additively, the mixture effect should not be underestimated, especially when we are mixing chemicals at really low exposure levels. According to the classical theory, similarly acting chemicals should be predicted by the dose addition principle, whereas dissimilarly acting chemicals should be predicted by independent action. However, experimental studies have told us something else. So mixture effects of dissimilarly acting compounds seems also to be that we can predict the effects of those by using the dose addition principle. And it has been shown here in vivo when we have mixed 4 anti-androgenic compounds and tested them for effects on anogenital distance in male rat pups. And it has recently been shown when 8 compounds were mixed to test for craniofacial malformations in zebrafish. And these chemicals, they acted by various mechanisms of action in these systems. And the mixture effect could be predicted by dose addition as well. And this is also good news for the risk assessor, as it makes things more simple that we can use. It seems that as if we can use the same mathematical prediction model to predict mixture effects of chemicals. When you do a cumulative risk assessment, you can base it on chemicals that either share the same mechanism of action, chemicals that target the same molecular initiating event in a specific AOP of interest, or you can use a disease of interest as the starting point. And I'll show you an example of both of these types of approaches. Here, the disease of interest was the starting point. It's poor sperm quality, and it's a really interesting study because it reflects true individual exposure levels. Usually we are using average aggregated exposure levels in a specific population, and that's what is available. But the cool thing here is that they have had access to individual exposure data in the same persons, and they've measured 29 chemicals monitored jointly in urine from 98 young men, and performed a mixture risk assessment which showed that there was a mixture risk and that the chemical mixture drivers were bisphenols, ASNF, some PCBs, and azalate, DHP. So in this case, human exposure data and rodent hazard data were used as the basis for the mixture risk assessment. Then an example of the other approach, where we have selected a molecular initiating event as the starting point. Here we have chosen androgen receptor antagonism as a starting point, and we did this case study as part of the HBM for Europe program. And our intention was to perform a mixture risk assessment based on human data only. So we identified more than 250 chemicals that are able to antagonize the androgen receptor. And for those chemicals, we looked for exposure data and ended up, after curation and so on, with 61 chemicals for which we had both reliable exposure and hazard data. And we calculated the hazard quotients of these 61 chemicals shown in this diagram, which also tells us which chemicals that are the drivers here. And the risk assessment ended up with a hazard index just below 1, and we found that the PCB 118, azalate, BBP, PFOS, and a UV filter BP3, benzophenone 3, were together contributing with 76% of the total mixture effect. And when we made this plot of the exposure as a function of the hazard data, we find the chemical mixture drivers in the upper left corner here of the figure. So I think it's important that we explore these new ways of performing risk assessment, and especially also involving in vitro data, as it'll allow us to make a more comprehensive mixture risk assessments in the future, as you know, toxicity data are often lacking for really many chemicals, as we've heard before. We have had a focus on developing tools for mixture risk assessment, and one of the tools is this user software interface called the Chemical Mixture Calculator, which is publicly available software that can be used for an initial mixture risk assessment. And we'll further develop this tool in our new Green Deal project, Panoramics. So this project, we started it almost 1 year ago, and we are 11 partners from 6 European countries, and it's coordinated by DTU, and it's will last for 4 years. And we are focusing on real-life mixtures and very much on in vitro testing of these mixtures. And yes, we have a focus on these complex real-life mixtures, and in the first year here, we have spent on testing extracts from either blood or water or fish or food samples in a large panel of in vitro tests. So our intention is to test for mixture effects across the environment, food, human continuum. And we have selected this large panel of in vitro tests that represent reproductive toxicity or neurotoxicity, which are the focus of our project. So in phase 2 here, we'll select a few assays that we will apply to human cord blood samples. And the intention is to determine the chemical mixture drivers in these various matrices. So, and this here is illustrated what we're going to do in phase 2, where we will perform effect-directed analysis on human cord blood samples from a specific cohort for which we have a lot of health information. So we will associate the chemical mixture drivers that we identify in the blood to health information on reproductive function or neuropsychological function. Yes, and I think my time has run out soon. The take-home messages are: 1 chemical at a time underestimates the risk. The dose addition principle can be applied for both similarly and dissimilarly acting compounds. And yes, we can predict mixture effects in most cases if we have adequate hazard and exposure data for single compounds. Usually we observe additivity in experimental systems, and synergism and antagonism is seen in rare cases only. There's a risk at high-end human exposures to certain chemical mixtures. And in the past, top-down designs have been performed a lot in mixture studies, but bottom-up designs will become more important for the future. And also we think that in vitro methods will be important for whole mixture assessments in the future. So with this, I would like to thank my collaborators, our funding sources, and also you for listening. Thank you.